So the lymphatic system functions to defend against infection and disease, and it returns tissue fluids to the bloodstream. Fluid is transported by lymphatic vessels called lymph. Fluid transported by lymphatic vessels is called lymph. Recirculation of this fluid is essential to maintain homeostasis throughout the body. Now, pathogens, these are disease-causing organisms. They include viruses, bacteria, fungi, and parasites. Each has different way of interacting with human tissues. The lymphatic system will include cells, tissues, and organs that defend the body. Primary cells of the system are called lymphocytes, and they are essential to the body's ability to resist infection or disease. Now, immunity, this is the ability to resist infection and disease. Immune response is the body's defensive mechanism against infectious agents and foreign or abnormal substances, such as transplanted tissues, chemicals, etc. Two forms of defense systems work independently and together: innate or non-specific defenses and adaptive or specific defenses. Now, your non-specific defenses, these are the anatomical barriers and defense mechanisms. What their job is to do is to prevent or slow entry of infectious organisms. Innate means that we are born with them. Non-specific means that they do not distinguish one threat from another. Specific defenses, on the other hand, lymphocytes respond to specific threats. The response to a present pathogen as well as future threats by the same type of pathogen. Adaptive, because the immune system will adapt and change depending on particular types of pathogens. This is called the immune response. So the immune system, all cells and tissues involved in the production of immunity. This includes parts of several systems, including the integumentary, which is your skin, skeletal, lymphatic, cardiovascular, respiratory, and digestive. All these systems will play a role in immune surveillance. So let's talk about the components of the lymphatic system. The lymphatic vessels, or lymphatics, will carry fluid from the peripheral tissues to veins. The fluid is called lymph. This flows through lymphatic vessels, similar to plasma but lower concentrations of proteins. The lymphocytes, these are specialized cells that function in defending the body. And then you have lymphoid tissues and lymphoid organs. Here are the various structures of the lymphatic system. Here we have our lymph nodes. You can see some ducts, some trunks, and then the various blood cells. Now, the lymphoid tissues, these are collections of loose connective tissue and lymphocytes. For example, your tonsils. Your lymphoid organs, these are more complex structures. They contain large numbers of lymphocytes connected to lymphatic vessels, and examples will include lymph nodes, spleen, and thymus. Primary lymphoid tissues and organs are sites where lymphocytes are formed and mature, and this includes sites such as the red bone marrow and the thymus, where red blood cells or blood cells, white blood cells, can form and mature. The B cells will mature there, and the thymus is where we see maturation of the T cells. Secondary lymphoid tissues and organs, this is sites where lymphocytes are activated and cloned. This would include things like the appendix, the spleen, the lymph nodes, the tonsils, and what we call mucosa-associated lymphoid tissue, or MALT. Now, the functions of the lymphatic system is production, maintenance, and distribution of the lymphocytes, the return of fluid and solids from peripheral tissues to the bloodstream, and the distribution of hormones, nutrients, and waste products from tissues to general circulation. The lymphocytes are produced in red bone marrow. They are stored in lymphoid organs, such as the spleen, and they respond to the presence of invading pathogens, abnormal body cells, such as cancer cells, and foreign proteins or tissues, such as toxins that might be produced by bacteria or even, mistransplanted blood or a transfusion reaction there. Your lymphatic capillaries, these are smallest lymphatic vessels. They begin as blind pockets in tissues. They are lined with overlapping endothelial cells. The region of overlap acts as a one-way valve. So what happens is as the fluid builds up in the tissue and that pressure builds up, the fluid will move into the lymphatics. But then as that builds up with inside the lymphatics, it closes the opening and the fluid moves in a one-way direction. Unlike the cardiovascular system, which is a closed circuit, lymphatic vessels are a one-way flow. Now, the walls of the small lymphatic vessels are very similar to that of veins. They contain valves to maintain one-way flow. Contraction of the skeletal muscles around the lymphatic vessels will aid flow, and lymph eventually empties into the collecting structures called lymphatic ducts. And the two ducts that we have are the thoracic duct and the right lymphatic duct. And here you can see those main ducts right here and here. The thoracic duct will collect lymph from the lower body, inferior to the diaphragm, and the left side of the upper body. The right lymphatic duct just collects the right upper side of the body, and it empties into the right subclavian. By the way, I'm sorry, the thoracic duct will also empty into the left subclavian. Now, T cells, these include your cytotoxic T cells, which will directly attack foreign cells or virus-infected cells. They provide cell-mediated or cellular immunity. We also have what are called helper T cells. This will stimulate other T cells and B cells, as well as suppressing those cells. It really regulates their function. Helper T cells are primarily what control your immune system. Helper T cells are what get attacked by HIV, and that's why people that have HIV that's not managed, what happens is the helper T cells will decline too much and you lose your immune function and you enter into what's called AIDS. The regulatory T cells, they will moderate the immune response, and memory T cells carry the memory of the agent that induced the immune response. Now your B cells, when stimulated by a specific antigen, which is anything the body perceives as foreign, will differentiate into plasma cells, and they will produce antibodies or immunoglobulins. They are responsible for antibody-mediated immunity or humoral immunity. Antibodies bind to specific targets called antigens. We have natural killer cells. These provide innate, non-specific immunity. They attack foreign cells, which are cells normally infected with viruses and cancer cells. They will continually monitor peripheral tissue as part of immune surveillance. And here we can see the origin and distribution of lymphocytes. Again, they're all developed in the bone marrow. B cells and natural killer cells will mature within the bone marrow and then go into circulation. The T cells will be formed in the bone marrow, but they will mature in the thymus and then go into circulation. And here we can see our natural killer cells with immune surveillance, our B cells with antibody-mediated immunity, and then our mature T cells, which is cell-mediated immunity. This is actually a very nice picture. I think I'm going to steal this from my micro class. So the lymphoid tissues, these are densely packed lymphocytes in loose connective tissues. They protect epithelia in body systems open to the external environment. Collections are referred to as mucosa-associated lymphoid tissue, or MALT. It is extremely important in the digestive tract. Examples include the tonsils, appendix, Peyer's patches within the small intestinal wall. The lymphoid organs, these are separated from surrounding tissue by fibrous connective tissue capsules, and this includes your lymph nodes, your thymus, and your spleen. The lymph nodes, these are small oval lymphoid organs covered by fibrous capsule. Multiple nodes are grouped together. The greatest number are located in the neck, armpits, and groin. The afferent lymphatic vessels will bring lymph to the node. The lymph node filters pathogens from lymph, and what happens is the phagocytic cells will then destroy the pathogens, at which point both T cells and B cells are activated, and then the efferent lymphatic vessels will drain the node. And what happens is fluid comes in, and then it goes through the node. It goes in through these 4 different areas, and then it leaves through this one area. The fact that you only have one exit to these 4 means pressure builds up, and the lymph will spend some time in here before it eventually goes through and filters out. Now the thymus, this is the site of T cell production and maturation. It's located in the mediastinum, which is your chest cavity. Develops to a maximum size during puberty, and then it starts to decrease in size. Remember, from you know early childhood all the way up through teenagers, that's when you're being exposed to the most things pathogenic. So that's why you're building up most of your immunity during those years. As you get older, past puberty, things start to atrophy, and you're not developing as many new antibodies to things, just because you've been exposed to a lot of things already. The thymosin hormones are made by the thymus, and they stimulate lymphocyte maturation. Your spleen contains the largest collection of lymphoid tissue. It functions in filtering the blood instead of lymph, removing abnormal blood cells and components. It initiates stimulation of B and T lymphocytes. It stores iron from the recycled red blood cells. It's located between the stomach, left kidney, and the diaphragm. Areas are red pulp, which contains large numbers of red blood cells, and areas of white pulp, which is where you have most of your lymphoid nodules. Now, looking at the body's defenses, there are two general categories of the body's defenses that work together to provide resistance. The first is innate or nonspecific immunity. It's genetically determined, it's present at birth, and it's nonspecific defenses. Your adaptive or your specific immunity is not present at birth. It's specific responses to an antigen. So looking at your innate defenses, they do not distinguish between threats, and they include physical barriers, phagocytes, immune surveillance, interferons, complement, inflammation, and fever. Let's talk about some of these in more detail. The first is physical barriers. They keep hazardous organisms and materials outside the body. Your skin is very good at its job. The keratin coating, layers of cells, and desmosomes junctions really act as a very strong barrier. Hair acts as a barrier to hazardous materials and insects, and secretions from glands will flush the surface and contain destructive enzymes called lysozymes and antibodies. The mucous membranes, this is another barrier that lines most surfaces inside the body, are found along the digestive and respiratory tracts. They're coated with mucus, they're flushed with glandular secretions, and they're aided by special enzymes, antibodies, and has a very acidic pH. With your phagocytes, these are cells that can surround and engulf solid objects. It is the first line of cellular defense. It will remove cellular debris and microorganisms, and it moves into tissues by squeezing between the capillary endothelial cells by a process called diapedesis. It is attracted or repelled by chemicals through chemotaxis. Neutrophils and eosinophils will leave the bloodstream and enter infected tissues to phagocytize. Neutrophils are dominant phagocytes within blood. Immune surveillance, this is the constant monitoring of normal tissues. It primarily involves natural killer cells. The plasma membrane of abnormal cells have non-self or foreign antigens. The natural killer cells recognize foreign antigens such as bacteria, viruses, and cancer cells. They respond very quickly, and they secrete perforins that create holes in the target cell's plasma membranes, killing those cells. And here's a summary of the body's innate defenses. You can see the physical barriers, the phagocytic cells, the immune surveillance, interferons, complement, inflammation, and fever. Again, this is another very nice figure. I think I'm going to steal from my micro class. Now, on the adaptive and specific defenses, this responds to the presence of specific antigens. It's provided by coordinated activities of T and B cells. Cell-mediated immunity or cellular immunity result of T cell defenses specifically against pathogens inside living cells. Antibody-mediated immunity or humoral immunity, this is the result of B cell defenses specifically against pathogens that are located in body fluids. Now, some features of the adaptive immunity: it's not present at birth, it can be active or passive, and it's acquired either naturally or artificially. Looking at active immunity, this is developed after exposure to an antigen, the result of the host's immune response to that antigen. Naturally acquired active immunity is due to exposure to pathogens in the environment. So this is somebody sneezing in your face and then you getting sick as a result of that. Artificially acquired active immunity. This is antibody production simulated through the exposure of antigens under a control condition. This would be what vaccines do. They are giving you the antigen and you're producing antibodies in response to that. With passive immunity, this is produced by transferring antibodies or lymphocytes to a person from another source. Naturally acquired passive immunity, antibodies provided to a baby through placental transfer through breast milk. And then you have artificially induced passive immunity. This is when antibodies are injected to fight the infection or disease. Example of this is antibodies given to protect against exposure to rabies or other viruses. So if you've ever gone for a rabies vaccine, they're essentially giving you antibodies to sort of combat that. Some properties of adaptive immunity, it's very specific. Specific defenses is activated by a specific antigen. Receptors on the plasma membrane of each T and B cell only bind to one specific antigen. It's very versatile. Millions of different types of lymphocyte populations are produced, each with different antigen receptor, additionally variable in antibody structure. It has memory. First exposure triggers development of memory cells. Second exposure to the antigen triggers faster, stronger, longer lasting immune responses. That's why vaccines work. You know, we give you the vaccine during a period of time when you are on your timetable, and then you develop immunity, that primary immunity, very slowly. And then when you're actually exposed to the antigen, it can take a very fast turnaround time to produce antibodies. Tolerance, it exists when the immune system does not respond normal or self antigens. Any T cell or B cell against normal antigens are destroyed during differentiation, meaning you can't have your own immune system attacking your own cells. So this is an overview of adaptive immunity. Again, we have the two branches, cell-mediated and antibody-mediated, and then it sort of gives you the little pathways down from there. So what are the 6 functions of antibodies? Why are they important? Well, they neutralize. This is an antigen-antibody complex that prevents the antigen from attaching to a cell. They can precipitate or glutinate. This is when multiple antigens can be bound with multiple antibody molecules, all attached together. Precipitation occurs when antigen-antibody complexes settle out of solution, and agglutination occurs when the target antigen is on the surface of the cell, producing large antigen-antibody clumps. Activation of complement, this is binding with an antigen, changes the antibody molecule shape, allowing binding with complement proteins. Attraction of phagocytes, this is the antigen-antibody complex attracts phagocytic cells that engulf pathogens. Enhancement of phagocytosis, this is the coating of antibodies to make pathogens easier to phagocytize, called a process called opsonization. And then stimulation of inflammation is when antibodies stimulate basophils and mast cells, mobilizing nonspecific defenses. And then here we have just a summary of a lot of the cells that participate in the immune response. Neutrophils, eosinophils, mast cells, macrophages, dendritic cells, natural killer cells, cytotoxic helper T cells, B cells, reg T cells, memory cells. So, just kind of look over the functions of all these. I'm sure I asked questions on these various functions, so they're all defined here. And then finally, this is the last slide. It just sort of describes the primary and secondary response. If you guys get a vaccine, what happens is just your first exposure, you develop a very weak response. And that's fine, and it takes a very long time. But then what happens then is if, let's say you got your measles vaccine. Okay, so you have 6 months, you developed immunity against it. Now somebody sneezes in your face and you're exposed to measles, you immediately develop a response and your immune system will attack it well before you even become symptomatic. So that's the primary and secondary response. First time's always slow. Second, third, fourth, fifth, sixth time will be very rapid. And that is it.